Hello everyone, this is Andrea Cabibbe from the San Raffaele Hospital. This presentation addresses the implementation of next-generation sequencing into TB laboratories for diagnostics or surveillance use. When implementing NGS for M tuberculosis, the strategy must reflect the local TB and drug resistance burden. In high-burden settings, there is a greater need for real-time prospective diagnosis of drug-resistant TB, while in low-burden areas, NGS may also be more focused on surveillance. Decision between whole genome sequencing from cultured isolates versus targeted sequencing from respiratory samples depend on the need for depth versus speed. Targeted NGS indeed offers faster turnaround time, often within days, and can bypass culture, but may provide less comprehensive data, for example on outbreak analysis, essential in low incidence settings aiming at disease elimination. Platform choice also influences turnaround time, workload, and cost. Benchtop sequencers are better for decentralized or lower throughput targeted workflows, while high throughput instruments are cost-effective for centralized WGS but require higher sample volumes and technical capacity. Cost considerations include not just reagents and equipment, but also bioinformatics, data storage, and staff training. NGS should initially complement rather than replace the other WHO-endorsed diagnostics. Crucially, results must be integrated into existing electronic systems with clear, clinical-friendly interpretations to guide timely treatment decisions and also public health investigation. The World Health Organization has developed several key documents to guide the implementation of NGS for drug-resistant TB since 2018. The WHO also provided catalogs of mutations associated with drug resistance, helping standardize interpretation across laboratories, which are described in a separate presentation. The latest edition of the WHO Consolidated Guidance about TB Rapid Diagnostics, released in 2024, include new recommendations on the use of a new class of diagnostic technologies, targeted next-generation sequencing tests for the diagnosis of drug-resistant TB. The recommendations provide a novel approach for the rapid detection of drug resistance to new anti-TB drugs using the latest technologies available, which is described in another presentations from this course. In 2023, the WHO released the NGS Implementation Manual. This comprehensive guide provides practical steps for national TB programs to adopt next-generation sequencing technologies, particularly whole genome sequencing and targeted sequencing, for monitoring drug resistance in M tuberculosis. The manual outlines essential components such as laboratory infrastructures requirement, quality assurance protocols, and bioinformatic workflows. It emphasized the importance of integrating NGS data into existing surveillance systems to enhance the detection of drug-resistant TB strains. Additionally, the document discusses considerations for resource-limited settings, including cost-effective strategies, training needs, and partnerships to facilitate technology transfer. By following these guidelines, countries can strengthen their TB surveillance capabilities and contribute to global efforts in combating drug-resistant tuberculosis. The WHO Implementation Manual outlines 10 essential steps for integrating NGS into national TB programs. First, define the intended use of NGS in line with the national TB strategies, considering objectives like surveillance or clinical diagnostics. Establish a technical working group to lead planning, perform readiness assessments, and oversee regulatory compliance. Select and set up appropriate equipment based on the intended use, ensuring safe and functional testing sites. Develop procurement and supply chain procedures to maintain a reliable flow of quality-assured reagents and consumables. Create comprehensive standard operating procedures, covering all aspects from sample collections to result report. Implement robust data management systems, including secure storage, bioinformatics tools, and confidentiality protocols. establish a quality assurance program encompassing quality control, proficiency testing, and regular performance monitoring. And last but not least, integrate NGS data into existing surveillance systems, updating forms and registers for seamless data capture and interpretation. The implementation manual includes 17 annexes that provide practical tools and resources for programmatic implementation. These annexes encompass implementation planning tools, templates like a Gantt chart and high-level checklist assisting in planning and tracking NGS implementation activities. Situational analysis resource, including checklists and real-world examples such as South Africa experience, guide the assessment of existing capacities and needs. Budgeting and procurement, guidance on budget considerations and lists of essential equipment and reagents, supporting financial and logistical planning. Information on commercially available NGS instruments and installation checklist, and aid in laboratory preparation. Estimates of data storage needs based on anticipated workloads help in planning for bioinformatics infrastructure. Key quality indicators and control considerations and details on proficiency testing programs ensure reliable results. Example of data and quality indicators and reporting forms facilitate standardized communication of findings. Simple terms of reference for key personnel, suggested training agendas and competency assessments support workforce development. Impact measures provide metrics for assessing the effectiveness of NGS implementation. These annexes serve as a comprehensive resource to support countries in the effective adoption and integrations of NGS technologies for TB surveillance. This table lists the essential equipments and reagents that are required for the NGS workflow, including general instrumentations and reagents kits for DNA extraction, library preparation, and next generation sequencing reaction. In this slide, the terms of reference of the laboratory personnel who is involved in the NGS workflow are presented, including a senior scientist, laboratory technicians or molecular biologists, and the bioinformatics officer. In this slide, I want to share the key quality indicators and quality controls considerations for the NGS workflow. You can appreciate that these are set from the beginning of the procedures, DNA extraction, through library preparation, sequencing, and then also for the bioinformatics part, with regard to the assembly, variant calling, and interpretation. I want to bring now a practical example of country implementation of NGS. This article presents a comprehensive account of introducing TNGS for diagnosing drug-resistant tuberculosis in low-resource settings, and specifically detailing a pilot program in Namibia. The strategy for the implementation of NGS was based on 3 pillar phases: preparation, implementation, and sustainability. These phases were subdivided into smaller categories and tasks. Key components include establishing appropriate laboratory infrastructure, developing SOP, ensuring quality assurance, and training personnel. The model advocates for a stepwise implementation of this capacity. Several obstacles were identified during the implementation, starting from technical issues, so initial difficulties with sequencing equipment setup and maintenance, challenges in establishing dedicated laboratory spaces and ensuring stable environmental conditions, delays in procuring equipment and reagents, compounded by supply chain disruption, need for specialized training for laboratory personnel, and clinicians also to interpret and utilize sequencing data effectively, and then ensuring secure and efficient handling of large sequencing data sets in terms of data management. Despite these challenges, the pilot successfully established a functional TNGS workflow in the Namibian National TB Reference Laboratory. Early results demonstrated that TNGS can provide rapid, comprehensive drug resistance profile, enabling more informed treatment decisions. This experience highlights both the feasibility and the complexity of introducing NGS into routine TB care in low-resource settings. As mentioned in the example previously, implementing NGS for drug-resistant TB in low-resource settings comes with several challenges. First, infrastructure is often limited. Labs may lack stable power, temperature control, and appropriate biosafety conditions. Procuring equipment and reagents can be slow and unreliable, with frequent supply chain interruptions. Human resources represent also a barrier. There's a shortage of trained staff in molecular biology, sequencing, and especially bioinformatics. Data management is also difficult with limited storage, computing power, and secure systems. Maintaining quality assurance is challenging without established protocols and access to external quality control. Costs are high, not just for the instruments, but also for ongoing supplies and maintenance. On top of that, regulatory hurdles can delay approvals and integration into national programs. Finally, NGS results often are not well integrated into care due to slow turnaround times and limited clinical training in interpreting genomic data. Timely access to testing should be guaranteed to people with MDR-TB, including in large countries where sequencing testing is centralized. And in this slide, I report the experience coming from several countries and the related reference to the published manuscript. Take-home messages from this presentation. NGS is one of the strategic technologies for the future. And actually, it is already used for real-time pathogen sequencing for diagnosis and surveillance. We need to couple long-term investment from the national programs with the sustainability plan. NGS is the only existing strategy for a fast screening for large number of anti-TB drugs, including the new drugs used in the MDR regimens. It is not simple to implement, but it is feasible. And the algorithms should be designed to use or upgrade existing laboratory services and networks. However, it requires clear and agreed strategy, algorithms positioning, budget, and political support. I conclude this presentation with a list of questions that I would like to address then during the live Q&A sessions. So for example, how do I choose the right sequencing technologies for my samples? We have seen in the presentation on the overview of the sequencing technologies that there are different chemistries, different sizes of the instruments, and all these are relevant if we want to perform NGS effectively in our TB laboratories. What is needed for NGS? And we will see a checklist for NGS implementation. And we have seen already in this presentation that the requirements for building the NGS infrastructures are quite high, in particular for certain sequencing technologies. And another questions we have is why NGS is better for drug resistance and outbreak analysis compared to other conventional or rapid technologies that are used for TB diagnosis and surveillance. Thank you very much for your attention.